And our next speaker is Dr. James McNally from University of Michigan. Dr. McNally is a research scientist, Inter-University Consortium for Political and Social Research, Institute for Social Research, Sociology, Demography, Brown University. His research focuses on aging and life course issues and on the methodological approaches to the improvement and enhancement of secondary research data. He's currently working on innovative tools and approaches to allow more efficient access to restricted data in a manner that maximizes both efficacy and subject confidentiality. He's currently lead to the NACDA. Now, I'm not sure what it is, which you may be able to explain, program on aging and the data achieving distributing studies on health and aging life courses. Today, his talk is going to be creating increased research interoperability between ADRD studies and measures of cognition. Dr. McNally, please.
Thank you. Second one down.
This one, right?
Yep.
And you can control it using this or mouse. Or
Okay. Let's see. Figuring this out real quick. Maybe I'll use the mouse then. Good morning. For something completely different. This has got nothing to do with clinical practice, genetics, or clinical options. But what we're trying to do is organize research data so people can compare and examine related studies, particularly in the area of AD and ADRD. I've been working on these concepts for over 30 years, and we're finally getting some traction. So that's part of the fun. Current funding, NIA owns me. They pay for everything from graduate school to my current research interest. Main things I'm doing right now is factors in aging, which the primary project is a major data archive that has multiple studies on aging in the life course. We've also got a R21, which is a phased award. We're hoping to move to a R33 in another year to develop these interoperability tools. We also just began a new COVID coordinating center at Michigan, and at the present time I'm the project director. Got a few other things going on, but I don't have any conflicts. I'm a social gerontologist. I love old people. I work with old people. And the great thing about being a social gerontologist is it allows me to float. It allows me to engage in team science. I can serve on faculty patient advisory boards at the UM medical system. I can work with sociologists. I can work with demographers. I can work with clinicians. I can even occasionally work with economists. I've done a variety of different things over the years, worked for CDC for a while, but I've been at NACDA since 1998. And because I work at ISR, which is soft money, I constantly beg for money. So if anyone has any money they want to give us for research, hey, we're happy. NACDA, this is the main project. It's a collection of about 1,600 data sets specifically focused on aging, gerontology, and the life course. It's been continuously funded since the 1970s by NIA, and it's part of ICPSR, which is the largest data repository in the world, arguably, about 16,000 plus studies. So we concentrate on aging issues. We have different archives with different topical expertise. As you can see, dementia and cognition issues really stand out on things that people are interested in using our data for. Everything we have is indexed and searchable, so on and so forth. We also have a code repository. So if you're doing something really clever with data and would like to share your code for validation, but also replication work, we encourage you to do that. That's great stuff. All right. This has been going on for a long time. And in order to achieve interoperability, this ability to compare studies and variables in a wide format instead of a long format, has been going on for years. And there are some precursors that had to be accomplished before we could move into this new phase of interoperability we've been working on for the past couple of years. This all goes down to superpopulation theory. I ran into that rabbit hole when I was in graduate school, and it sort of consumed me ever since. The idea of a superpopulation is whenever we draw a sample, whenever we draw a group of test subjects, we're defining a finite population as part of the infinite population, the superpopulation. So again, in theory, we should be able to treat all data sets as part of a continuum, as part of a single data set. If we know the appropriate distribution. The Bayesians love this. But what I want to do and what I've been working hard to do is to treat my collection as a single data set where people can come in and pick out what they need. It may be studies that span 20 or 40 years. It may be comparing the way people measure specific variables. It may be almost anything. But I think of my repository as a single data set containing information that goes back to the slave records that Chicago originally collected to the most recent data sets. It's all there. How we put it all together, still working on that. What it requires is variables and metadata. These are the most basic and important aspect of achieving interoperability. Archival best practices are essential. We need to standardize the way we treat our data. We need to standardize the way we share our data. This has been going on since I got to ICPSR in the 90s, and I expect it will outlive me by decades. There is so much work to be done, but we're getting there. It's really exciting. Metadata is data about data. Every study in NACDA, every study in ICPSR has a metadata record. And it provides summary information about the study, provides data and documentation about the study, it provides information about the variables in study. And it also gives you a literature review of people who have used that particular study. In this case, Dr. Peterson's study has been downloaded about 50,000 times since she first gave it to us. This is also associated with Margie Gatt's work on dementia. She's drawn a subsample to look at dementia among twins. We have that data. Variables. We have to define each variable. Every variable that everyone collects, we want to define and be able to do that at the value label. So we can directly compare how you're measuring something, how you're measuring something, and how you're measuring something. Is it similar? Is it completely different? Can we harmonize it? What can we do with it? So far, we've identified about 6 million variables at the value label. ICPSR in general has a little over 16,000 studies, and we've identified well over 100,000 publications that use the data in the collection. Searching at the variable level is nice. So for our purposes, if we put in the word cognition, into the study field. We find that we've got a little over 2,000 studies that address issues of cognition as part of their study design. We've got over 4,000 variables that in some way or another touch on cognition. There's series data, there's publications, etc. What happens down here is we get a long list of variables instead of the wide list that I prefer. And if you were to click on that, it would take you to that variable and let you see what the values are. So that's cognition, a little broad. If we go to Alzheimer's, it narrows down considerably, 621 studies that deal with Alzheimer's, about 2,000 variables, same deal. You get a long list of those 2,000 variables, stacked one after the other, you can look through them. That's part of the process. This is where we were a couple of years ago. And it's the important, you have to do this before you can move on to interoperability. Interoperability, and this is the formal definition, but basically what it means is we array information in a wide format. So the researcher can look across multiple waves of a longitudinal study, across multiple studies that are independent from each other, and identify commonalities and variables. That is interoperability in our world. Now, make this work, we have to have consensus. We're working with what is called common data elements. And some people, particularly if you're in the biomedical field, you've probably heard of common data elements. Other people have not. Basically, it's a comparison list of variables that researchers have validated and accept as good variables. They're standardized key terms or concepts. Had some nice conversations with Mike Spittle over at NIH or OBSSR. He funded a little of our early work. And his goal is to do the same thing that the psychologists and the psychiatrists did with DSM, create standards, just have a giant consensus among topic experts in Alzheimer's, in genetics, in social science, et cetera, who agree what means. As a demographer, we're going through this now with gender. Male and female is not acceptable anymore for research because we recognize gender fluidity, we recognize different choices. How do we measure that effectively? And how do we go back and adjust for the past? So these are the kinds of issues that we deal with, but if we can line this information up, we can start making intelligent decisions. Okay, so how's this consensus going? Not so good. All right, right now, NIH is currently funding a little over 7,000 studies on cognition issues. As you can see, NIA is the big player. Mental health and neurology are the second and third in that group, with a variety of other institutes funding different resources. So NIA is really the driver of cognition research. In terms of Alzheimer's, it's even a bigger disparity. NIA is more than half of the 7,000 studies that are addressing Alzheimer's specifically. Neurology is the second, mental health drops to third. A lot of research going on out there. Publications. There's about 80,000, 81,000 publications that are associated with these currently funded projects. Again, NIA, big, mental health, big, neurology, big. NICHD is starting to pop in here. So these are the publications, the spread of publications. All these publications have variables. They measure things in a specific way. We would like to know how they measure it across different publications and studies, so we can see if they're measuring the same thing. As the people who do biomarker work, assays, and the way information is processed in the lab can have a huge influence on the results. All right, so NIH curates about 30,000 common data elements right now on their website. Neurology is the big player here. The next cancer, etc., heart, lung, and blood. They all are big players in the common data elements world. You'll notice a player is missing. NIA is not actively involved in common data elements. Now, of the 29,000 common data elements, there's only less than 3,000 agreed upon common data elements for cognition. And again, neurology is a big player there. Heart, lung, and blood, secondary. NIA is not in the picture at all. Alzheimer's, neurology, a couple other players. NIA is not there. NIA is not working to develop a collection of common data elements that are agreed upon. And we want to address this. So this is what we're doing now. We want to start organizing NIA-funded data within a common data elements framework, so they can also be part of this opportunity. We did a few proof of concept projects over the past couple of years. We use what's called the DDI XML standard. Don't worry about it. It's just a markup language. I don't do it. I have people who do it. But what it does do is let us manipulate repeat measure data, regardless of where it comes from. It can be the same multi-wave study. It can be totally different studies, but it allows us to organize it in a standard way that makes it searchable and discoverable. We started the DDI thing years ago, and we continue to work on it. This flavor is DDI 3. What's nice about DDI 3 or DDI lifecycle is it allows us to employ the variable cascade. And what you want to think about this is a nested system of information where we go from the broadest concepts like AD, RD, and then we would break it down into dementias, into different kinds of Alzheimer's, into different neurological problems. We would identify the variables for each of these, and then we would look at the way these variables are measured. So it allows us to treat data big or small. It also allows us to arrange the data in a wide format. It's a circle. In order to do this, we need the variables. We need that list of variables. We can do nothing if we haven't done the early work of building the variable list and the metadata. If we have these variables, then we can create a crosswalk, and we can start looking at variables and see how well they match. This is an example of a concordance we did for a comparison of the NSHAP project in Chicago and the NHATS project in Baltimore and And we're not interested in harmonization. Harmonization is expensive, it's clunky, and by and large, only about a dozen to 2 dozen variables in any harmonized data set are actually used for research. What we're interested in is the distribution of comparability. Is it directly comparable? Is it a one-to-many comparison? Does it need more harmonization, or is it just related in some way? So we've created a distribution of comparison, so you can look and see how closely two similar variables reflect each other and make decisions accordingly. We store this in the Collectica portal. Collectica is a company in Minnesota, and they have, at the moment, the best software to deal with interoperability and DDI life course issues. The nice thing about it is it lets you select variables you like, download them in a basket, and do your research. If you find you missed something, come back 5 minutes later, download the variable you're missing, and continue. It's always open, it's always available. So the portal, we have what's called a cross-series comparison portal. We also have individual studies in our portal. This is an example of Cognition comparing NSHAP to NHATS. Over here, we have the main concepts. So if you click on any of these, it opens up another field. And basically what we've done is compare every variable in NSHAP to every variable in NHATS. So you can see, regardless of what you're interested in. If you're interested in cognition, well, you can see that self-rated memory, they're related, but we have them for all 3 ways of NSHAP and all 8 ways of NHATS. So if you click on any of these, you will see what the variable is, how it's measured, what the labels are. And you can decide, can I do a comparative analysis, or do I just want to get a few years from NHATS and download it? Other things, cognitive impairment, they measured it in NHATS, did not measure it in NSHAP. NSHAP asks questions specific to Alzheimer's and dementia, NHATS does not. So it gives you a feel for the science. Also provides guidance. Why, if you're going to build a new study, then hey, maybe you should be following one model as opposed to another. Now, same thing for memory. If we look at memory, we can see total today's month correct. And NSHAP started collecting in wave 2 and wave 3, and NHATS again collected across all 8 waves. So you can start to see how people are thinking about their designs, what kind of questions they're asking, and who's got a better way of approaching it. We've also done this for MIDAS. We're working on doing it for Ad Health and the Wisconsin Longitudinal Survey, which is going to be a bear because that is a huge study. But all of this information can be fed in, allowing you to look at multi-wave studies based on concepts, based on variables, and variable content. And you can compare very quickly across studies. Okay, so the pilot worked. We can do this. We're very excited about that. We need millions and millions of dollars, but we'll keep working on that. Next steps and cautions. So this is generally where we're thinking, at least on the behavioral social science end. Over the next 5 years, we'd like to introduce all of these studies and compare them across time. We already got MIDAS online. We're working with these 2 already. We've got, of course, collaborative partnerships with HRS, PSID. We already have NSHAP. We work very closely with NHATS. These 2 are great out in California. Lots of meat out there. But the point is, we can do this with any study. We can take any information you have. We don't need your data. We just need your variables and your value labels. And we can start comparing across studies. So biomedical, clinical studies, it's all possible. Of course, this is the problem with data. This is Wound Man. I was introduced to him when I was in forensic. And it's just all the horrible things you can do to a person. Data, same way. We've got to deal with sampling bias, underrepresentation, unvalidated questions, etc. The gentleman who did the talk on the minimal data set gave a great example of the caution you have to take when you're working with data. And we're working with hundreds and thousands of data sets. So everyone's got a different perspective, and we have to arrange this as well. But that's what we do. We don't want you to do it. We get paid by the federal government to do it. We want to help you use the data more effectively. Unfortunately, not everyone wants to share their data. Social science research, we've got a long tradition of sharing data. Epidemiologists have been very supportive over the past decade or so as they saw the payoff in terms of increased publications and increased use of their data. Psychology, we're making good progress. I'm not disappointed. Biomedical, we've made some progress. Clinical trials, still banging my head against that wall. But the rules are changing a lot in terms of data sharing and funded research. So we're preparing for a flood of clinical trials data, probably in the next 5 or 6 years. We need all this information if we're going to do population health research. We all have our specialties. We all have our little baskets for professionalism. If we stay in our baskets, we get nothing accomplished. We only get one side of the picture. I love team science. I love multidisciplinary research. I learned so much talking to clinicians and geneticists, and hopefully they learn something from me. But if we can all work together, then we can tackle this social determinants of health. And that is the key. That is the environmental, social, microaggression, all of the problems that a human being faces in their lives that affects why Larry has Alzheimer's or cancer and Bob doesn't. And this is huge. I love population health stuff. We don't have the data except in very small samples. Well, the small samples we do have show that we can show very different. University of Michigan, very interested in cardiac outcomes. People come from all over the country, all over the world for heart surgery. If you live in Michigan, you get continuous care. You get heart surgery and go back to Arkansas, you might have a very different outcome. It's not because of the quality of care, it's because of the social determinants you live with it. Yep, I'm going to call it. We can overcome. We just want your variables. We don't need your data. You can keep your data. You can decide who you collaborate with. This is a very important point with a lot of researchers. You can work collaboratively with others, but we want to publicize. We want to make it transparent. We want to make it discoverable. If you're doing something really exciting in Alzheimer's research, we want to make sure that all the other researchers know what you're doing and why it's important. And then they can come to you, work out some kind of deal. Also, stability in measurements. If we're not measuring things in the same way, we're measuring different things. So part of the interoperability is to allow people to compare what other people are doing and decide for themselves if that's the direction they want to follow, or if they have a better idea, consensus. We want to thank our partners and collaborators. These are all people who work directly with us on their data. These are people we work with on their data, and 1,500 more studies. I can be reached anytime. You can Google me. We have a website, we have Twitter, we have Facebook, we have email, and we're at NACDAaging.org. If we can help you.
Great talk.
Thank you.
We may have a couple of minutes for questions.
Or it may just be way too much to absorb in 30 minutes.
There are no new questions.
So just, you mentioned clinical trials. And the main question I have about it, what are the obstacles? You heard previous talk about so many clinical trials. Is it the industry which is holding that collaboration? Why? It seems like that was the main area where the data sharing was difficult. Please.
It's also culture. I mean, there's just not a culture of data sharing within clinical trials. They're looking for a specific outcome, and they're not really thinking about the ancillary studies that might emerge. Also, yeah, I've worked with the co-creating collaborative. I've done talks. There's almost an anthropological perspective that this is my tribe and I don't want to share it with anybody. The cigarettes studies, for example, all the people who got subpoenaed under the Shelby bill, and they were very worried that the cigarette companies would use their data to disprove their research. So there's also fear. It's a question of trust, and we need to build trust relationships. We don't want to steal anybody's research. We don't want to steal anybody's work, but we do want to make it transparent so people are aware of what's available and they can build partnerships and collaborations. Some things are wide open. Health interview survey, wide open, anybody can get it. A clinical trial, well, there's confidentiality and respondent protection issues. You may have to work a special relationship with the researcher. Those are all fine. Any data can be shared.
Then maybe you may answer one at least.
Okay.
From the chat, you can read it yourself.
Okay.
Here it is.
Let's see. Can you make the background
Oh, I'm
picture?
sorry, this one.
Oh.
This one.
I'll mention the ADRC UDS. The ADRC UDSs, it's mentioned. I think, I'm not quite sure what that is. So I stand to be educated on that.
There's another question here. Could you
Jim,
comment
it's
that?
a uniform database that
Okay, another one of those, yeah.
that all the Alzheimer centers collect, and there's a central coordinating center in
Mhm.
UW and Seattle.
See, that's part of the problem. You know about it.
Yeah.
How many people knew about common data elements before I mentioned it? This is why we need to share this kind of information. Now that I know about it, I'll promote it.
Yeah, there's also something called AMP-AD,
Mhm.
which is across several different spaces,
Mhm.
and well, the AMPs are several different spaces, but the AMP-AD also has a portal at Sage Bionetworks
Mhm.
and synapse.com.
There are literally hundreds of question databases out there, and they're generally just known to the professionals within that branch. With interoperability, again, it's team science. We want everybody to know about this. This is great information. We'll start promoting that as well. Let's see, what was the other? Could you comment about whether similar differences between observed between original and offspring cofix, if you compare them to any other US-based cohorts or to ones from other countries? Share is a good example of that. It's a multinational study of aging. It's one of the HRS sister studies, and they look at everywhere from Sweden to Israel, looking at different health and income outcomes. Gary somebody, I'm sorry, I'm blanking. Guy at Harvard, he's created what he calls vignettes to help explain cultural differences between the different countries to make them more comparable. But there's always going to be differences in those situations, and we just have to work with it.
Great, thank you very much. Thank you very much. Jeannie? We are running a couple of minutes late, but anyway, we continue to move on.